The title of my talk is Nanotechnology for COVID-19, a small step towards a giant leap. I'm Vandana, Professor of Pharmaceutics at Institute of Chemical Technology. And this is where I do my research, that's Institute of Chemical Technology, which is one of the premier institutes in India. It was established in year 1933 and till date has given 19 Padma awardees, which is one of the highest civilian awards in India. We are familiar with COVID-19 timelines, but just to recap, in December 2019, it originated from the city of Wuhan in China. WHO announced COVID-19 outbreak as a pandemic on 11th March 2020. And by 4th of April 2020, over 1 million cases of COVID-19 worldwide were detected positive. Looking at the structure of coronavirus, it has on its surface E protein, M protein, but most prevalent is S protein, which is short form for spike protein. Even the transmission electron microscopic image indicates the structure of these spikes. And it is with these spikes that it latches onto human cells and undergoes a structural change, allowing the viral membrane to fuse with the cell membrane. Viral gene then can enter the host cell to be copied, producing more viruses. Now, this particular spike virus is 10 to 20 times more likely to bind with these two on human cells than that of SARS virus of 2002. And in fact, that is the reason it spreads more easily from person to person than earlier viruses. Despite similarities, 3 different antibodies against the 2002 SARS virus could not successfully bind to this particular protein. This also indicates that there's a dire need that we come out with a potential vaccine or antibody-based treatment strategy, which is unique to this particular virus. WHO situation report in July 2020 has clearly indicated that there has been more than 17 million confirmed cases of COVID-19. And this has resulted in more than 6,70,000 deaths world over. As far as status country-wise is concerned, USA is with the highest number of confirmed cases, followed by Brazil, India, and Russia. Within India, there are certain states which are much more affected as compared to the others. So these include Maharashtra and Tamil Nadu. And in India alone, there have been about 1,695,988 confirmed cases of COVID-19, with 36,511 deaths from 30th Jan to 31st July 2020. So researchers are working on a variety of nano strategies at all levels, be it for prophylaxis or diagnosis or therapy. We'll see each one of them one by one. As far as prophylaxis is concerned, vaccines as well as surface protection techniques would come under prophylaxis. In the vaccines category, if one looks at it, we all are familiar with the concept, and we know that vaccines work by tricking our immune system into thinking that it's being attacked by a virus. Immune system then churns out antibodies that get home to that virus. So in case at a later date, if one gets exposed to that virus, body quickly squashes it out. So before it can actually make you sick. So as far as vaccine delivery is concerned, there are 4 technologies. So either one could use inactivated virus itself or use protein-based viral subunits, say S1, S2. People have also utilized adenovirus vector for delivery of antigen, because these are virus themselves. So it's very, very easy to get transfected inside the host. And then you have gene-based technologies, which include mRNA or DNA-based vaccine. Nanocarrier selection for antigens. Most commonly used one is PLGA nanoparticles because scale up for these is very well known. Others which are being utilized include gold nanoparticles, carbon nanotubes, dextran sulfate and chitosan polymeric nanoparticles. Nano has also been utilized as vaccine adjuvants, and we know for sure that role of vaccine adjuvant is to make the antigen more potent. Besides, it has to have good immunomodulatory capacity, antigen-specific clonal expansion, and it should also give long-lasting adaptive immune response. So historically, PLGA nanoparticles and gold nanoparticles, as I mentioned earlier, have been used, but people have also tried adjuvant co-loaded liposomes, antigen conjugated alumina nanoparticles, as well as certain oil and water emulsions, ESCOMs, virosomes, etc., which had given quite a success for influenza vaccine. And this slide depicts influenza vaccine adjuvanted flu ad, which is quadrivalent for adults 65 years or older. In fact, this contained MF59 adjuvant. And by addition of this adjuvant, led to strengthening of the immune response by stimulating more immune cells to create more antibodies. Also, it broadened the immune response by creating more diverse cross-reactive antibodies. If you look at the scenario worldwide, this is pipeline of COVID-19 vaccine candidates by technology platform. Exploratory projects are split into two, confirmed and unconfirmed. They are in the early planning stage, whereas there are some which are in the preclinical stage. Animal testing is going on, and then there are some which are entered phase 1. Irrespective of current stage of development, one can see that nano-based technologies are most prevalent for vaccines. If you look at profile of COVID-19 vaccine developers by geographic location, also one thing which is very clear is industry is very actively working in this direction, many a times in conjunction with academic institutes or public institutes. Even some non-for-profit institutes are working in this direction, all working with a single-minded goal to develop a vaccine so that world is free of this corona. DCGI has given permission here in India for the first and second phase of human clinical trials to Bharat Biotech International Limited, which is working in collaboration with ICMR for a vaccine, and Zydus Cadila Healthcare Limited. And these will be available as Covaxin and Zycov-T once successful. And as Mr. Pankaj Patel, Chairman Zydus Cadila Group, says, it's time for all of us to rally together and help one another so that we are able to overcome these challenging times. Draft landscape of COVID-19 candidate vaccines Indian scenario lists n number of vaccines at different stages. Most of them, of course, are in the preclinical stage, but there are some who are in phase 1 and phase 2. There have been some candidates which have been tried earlier for other viruses and they have been roped in for SARS-CoV-2. Whereas in some cases, totally new targets have been identified. As far as surface protection is concerned, we are aware that this virus gets inactivated if pHs are extreme. At pHs of less than 3, pH more than 12. Also gets inactivated by heat treatment above 65 degrees Celsius with high alcohol concentration, more than 80%, and then by use of UV. Utilizing these techniques, many people have come up with many equipments which can actually lead to deactivation of this virus. This slide just depicts how long the virus is viable on different surfaces. Nanotechnology-based interventions can also be used for this purpose to create self-disinfecting surfaces, biosurfactant coatings. One could have metal-based nanoparticles, which can act as intrinsic antimicrobials and antivirals. Then there are certain nanoparticles like titanium dioxide, which can act as photocatalytic. And if these could be coated onto masks, etc., or onto equipments and then given photo irradiation, virus definitely would get inactive. So a lot of these approaches are being tried in research and in hospital setups to see their utility back. The most prominent ones amongst the metallic nanoparticles are the silver, copper, and graphene. Silver ions are known to interact with surface protein of viruses. So they are envisaged to confer biocidal properties to films used in healthcare settings, air filters, face masks. Copper also has been utilized, in fact, nanoparticle form to impregnate masks. And graphene, because of its charge, can bind with positively charged virus. So graphene derivatives also have been linked to virus-specific antibodies, and a lot of research is going on in this direction. One Indian innovation which is worth mentioning is from Defense Institute of Advanced Technology, and they have developed a cotton mask utilizing the herbal extract. In fact, these herbals, many of them could be in the nano form. Non-woven nanofiber-based three-layered biodegradable mask named Pavitrapati already is in use. As far as diagnosis is concerned, we have majorly three techniques: molecular RT-PCR, antigen-based techniques, and antibody tests. All of them utilize either nasopharyngeal swab or oropharyngeal swab, serum, or plasma for testing purposes. Antibody tests will detect IgG, IgM, and IgA. Looking at each one of them in detail, overall the kit market is growing, and cumulative annual growth rate of about 17.3% is projected. By year 2026, it is more than $8.5 billion market. Potential for businesses there, but majorly diagnostics would based on tests will detect the virus like RT-PCR and antigen tests, and tests will detect immune response and potential immunity, which include the antibody tests. So as time and disease progresses, in fact, it's very important that both these populations are detected so that even asymptomatic cases could easily be mass screened and isolated. In India, we have number of diagnostic kits which have come into the market. Pune-based molecular diagnostic company MyLab Discovery Solutions came up with India's first COVID-19 test kit called as Therapeutic. A GAP Chitra Magna, a magnetic nanoparticle-based RNA extraction kit, was commercially launched in May 2020. Then there is a kit by ICMR based on ELISA, referred to as COVID-Kavach ELISA, and this technology has been transferred to Zydus Catala for mass production of kits. COVID-19 microELISA D is the first total antibody detection kit developed by J. Mitra and Company, Delhi. And then there is CoroSure, which is low-cost, probe-free RT-PCR-based kit developed by Newtech Medical Devices after obtaining license from IIT Delhi. So a lot of academia, industry interaction has come into play because of COVID-19. One particular nanodiagnostic kit, which I would like to mention, is based on gold nanoparticles, and it's an immunochromatographic test kit. Test is quite simple. Sample would be collected, would be added onto a cassette, followed by addition of a sample buffer or 3 drops, and test results are read after 10 to 15 minutes. So if you look at what would happen with the gold nanoparticles is, if the patient is corona positive, the SARS-CoV-2 virus is present in the swab, it will interact with gold nanoparticles. Net result would be these nanoparticles would get aggregated, and that would result in change in color. So it's a visual change in color which can be seen. So with naked eye, detection of COVID-19 would be possible. So sensitivity of this nanodiagnostic technique is much more as compared to the others. Another interesting research which is happening is with respect to nanopore target sequencing. This confirms SARS-CoV-2 infections within minutes by mapping the sequence to the SARS-CoV-2 genome and analyzing the identity, validity, and then reading the number of output sequence. So this kit has come out as Nanopore Technologies, Oxford Nanopore Technologies, and the video which you would see now very clearly would indicate how this technique is highly sensitive. So it is available as a small box wherein it actually contains a flow cell, just like lateral flow kit. So you do have DNA, RNA sensors. Just a drop is sufficient to conduct this test. There are about 2048 membrane wells. So it's a very, very quick test. You see, this is a nanopore. So with the tether, it gets into contact with the nanopore. And typically when this happens, very, very high charge density would be produced, because of which disruption of ionic current happens, which is measured in signal trace very quickly, resulting in measurement of a very, very small nano, or rather pico amounts of virus present. So this particular technique is being utilized by Nanopore Technologies, as mentioned earlier. As far as therapy is concerned, India is having know-how for almost all the drugs which are mentioned here. So be it hydroxychloroquine plus azithromycin, which was the initial line of treatment, or antiretrovirals including lopinavir and ritonavir. A lot of emphasis is also given on adjunctive therapy like vitamin C, vitamin D, and zinc, etc., as immune boosters. And then we have dexamethasone, and now, of course, remdesivir. Mesenchymal stem cells also are being researched on for treatment option. If you look at therapeutic targets for COVID-19, very clear that remdesivir inhibits viral RNA-dependent RNA polymerase, and therefore it has its potent action. But besides that, there are others like CQ and HQ, which inhibit glycosylation of host receptors to block virus entry. Government of India has given a lot of stress on repurposing strategies. And the major targets which are being looked upon are viral proteases, host cell produced proteases like TMP, RSS2, RNA polymerase, or interaction site of viral S protein with host receptor, that's ACE2. In fact, Ministry of Ayush has given immunity promoting measures as guidelines, which include utilization of actives like turmeric, besides applying sesame oil or coconut oil or ghee in both the nostrils, or oil pulling therapy, etc. What's very important is phytochemicals which are being screened majorly belong to these categories: phenolics, alkaloids, carotenoids, terpenoids, and nitrogen containing compounds. Molecular docking is leading the way. In fact, there is a hackathon which is undergoing which has promoted all students and researchers to look at molecular docking of many of the known actives and repurpose it for COVID-19. And some of them are mentioned here. So monoterpenes, eucalyptol, curcumin, sylvestrol, tomentine, as well as luteolin, nicotinamide, epigallocatechin. Even our lab has worked on epigallocatechins, gallet, and proven in silico that it has activity similar to remdesivir. People are also researching quercetin, glycyrrhizin, and coating the nanoparticles with lectins so that fusion with the host membrane becomes easier. This slide summarizes the formulation strategies. So if one has to come with COVID-19 nanomedicine, it has to address the therapeutic challenges of the known drugs, which include poor aqueous solubility, lower bioavailability, systemic toxicity, because you are dumping more amount of the drug in, and problems which are related to shorter half-life. So repurposing strategies which are being employed are chemically altering or re-engineering the drugs for a particular nanocarrier, or selecting particular nanocarrier for a particular drug. It could also be nanomedicine for combination drug therapeutics. Of course, vaccine development is totally a different story, which we have already talked. As far as opportunities for nanotechnology are concerned, therefore, we need to look at whatever nanocarriers earlier we have fabricated for BCS class 2 and class 4 drugs, and see how the drugs which are repurposed can be incorporated using those techniques. So be it for increasing solubility and therefore bioavailability, which then results in dose reduction and reduction in side effects, or it could be for prolonged action or control release purposes too. So broad range of active moieties, including antiretrovirals, biologics, and nucleic acids, can be co-loaded and delivered. So co-encapsulation of both hydrophobic and hydrophilic drugs is possible. For example, plasmid DNA along with chemotherapeutic drugs could be co-loaded. This definitely will improve the therapeutic index. Of course, one needs to look at preserving synergistic drug ratios, overlapping pharmacokinetics, and therefore reducing the combination alike side effects. Once this is done, I'm sure that nanotechnology based strategies would definitely come up with a therapeutic option for COVID-19. People are researching on PLGA nanoparticles to increase efficacy and reduce toxicity. Lipid nanoparticles, wherein siRNA has been loaded, lipid-coated mesoporous silica nanoparticles, where delivery of hydrophilic and unstable molecules is possible, and nasal and pulmonary routes of delivery also being exploited using nanocarriers. In fact, when our lab is working into this area, direct delivery to site of action. Chemically altered or re-engineered drugs, one has liposomes, which could be loaded both by amphiphilic and ionizable drugs. In fact, HCQ loaded liposomes, which are modified with cholesterol, already have been tested and they've shown lower dose as well as toxicity. Prodrug synthesis is one area where very critically one needs to look at. Nanomedicine for combination drug therapeutics. So nanosuspensions could be made of all the drugs which belong to BCS2 and BCS4, and then multi-drug loaded pegylated liposomes definitely can be manufactured for prolonging their circulation in blood. Plethora of possibilities therefore exist using nano, be it use of nanomaterials in PPEs, personal protective equipment, or neutralization of viral particles by nanomaterials, nanomaterial-based photodynamic therapy, or nanomaterial-based vaccines and immunomodulation. In all the cases, either it could lead to systemic inactivation of SARS-CoV-2 or it could lead to activation of immune system. As mentioned earlier, delivery of nanomaterials to pulmonary system is also possible, and on-site targeting ACE2 receptor or viral S protein might give perfect answer therapy of COVID-19. So in conclusion, COVID-19 global emergency poses unprecedented challenges. Our primary health needs must be addressed, having in mind one health concept. Anyone who comes up with the therapy should make it immediately available worldwide. So for this, scientists from diverse backgrounds need to converge in fruitful cooperations for catering to such pandemic. And nanotechnology is definitely needed to lay new foundations towards counteracting current global public health threat. Thank you so much. But till we have any therapy or vaccine, we need to break the chain of COVID-19 transmission. And this is picked up from WHO. This is thank you from my nano group, and I would like to end the presentation in words of Stephen Hawking. Intelligence is the ability to adapt to change. So we need to really get adapted to newer delivery systems as fast as possible to tackle this menace. Thank you so much.